Imagine for a second having a tiny microscopic USB drive that you could just plug directly into your body's immune cells.
Right, just a direct biological upload.
Yeah, exactly. You just upload a new software patch and instantly, you reprogram them. You transform these cells from aggressive tissue-destroying attackers into, well, peaceful tissue-repairing healers.
Which sounds like science fiction. I mean, it really does.
It totally does. But today, we are doing a deep dive into the exact biological mechanism behind that switch. We're looking at exosomes, which are basically these biological USB drives that hold the key to curing acute lung injuries, stopping rheumatoid arthritis, and even, and this is wild, dismantling the defenses of a tumor.
That is arguably, the most dynamic area of modern immunology right now. We're looking at the precise mechanisms that dictate macrophage polarization.
Right.
Basically how a cell is sourced from a pro-inflammatory M1 phenotype straight over to a reparative M2 phenotype, and it's all done entirely through exosomal signaling.
And, the stack of scientific literature we are pulling from today goes deep into the tumor microenvironment, acute respiratory distress syndrome, or ARDS, and some just insane advances in extracellular vesicle engineering.
Yeah, the engineering side is mind-blowing.
It really is. So our mission today is to decode the how and the why of the cellular hijacking. But I guess to start, we really need to look at the machine being reprogrammed, right? The macrophage.
Yeah, you have to know the baseline before you can understand the switch.
Right. And I tend to visualize macrophages as like the Pac-Man of the immune system.
Yeah.
Just roaming the tissues, devouring debris and pathogens.
I hear that Pac-Man analogy a lot, actually. But it kind of undersells their complexity, I think.
Oh, really? How so?
Well, they aren't just roaming, mindless eaters. They are highly plastic. They fundamentally alter their functional state based on environmental cues. But, for the sake of the two extremes, we usually look at the M1 and M2 states.
Okay, right. So break down the M1 state for us.
Yeah. So the classically activated M1 state is basically your scorched earth defense. When they encounter pathogens, they just blast out pro-inflammatory cytokines, things like IL-12 and TNF-alpha, alongside heavy reactive oxygen species.
Like they're literally throwing biological grenades into the tissue.
Exactly. It's highly effective for clearing an infection quickly, but, there's always massive collateral damage to the surrounding healthy tissue.
Which is why the M2 state is so critical, right?
Yes, exactly.
Yeah.
The alternatively activated M2 macrophages, they secrete IL-10 and TGF-beta. They are the ones that initiate tissue repair, matrix remodeling, and they basically start rebuilding the local architecture that the M1s just blew up.
Okay, so bringing exosomes into this. These are the extracellular vesicles, right? Like these tiny lipid bubbles packed with proteins and nucleic acids that travel between cells carrying genetic instructions.
Yep, they are the messengers.
So if an exosome delivers a payload that forces a macrophage from M1 to M2, it stops the destruction and starts the healing. And just listening to that, it sounds like, well, M2 is the unequivocal hero of the story and M1 is the villain. Like, we always want to force that M2 switch, right?
Well, what's fascinating here is that biology completely ignores our concepts of heroes and villains. Context is just, it's everything.
Okay, how do you mean?
So in the case of rheumatoid arthritis, yes, you desperately want M2 macrophages to stop the joint destruction. But in oncology, tumors actually completely hijack this system.
Wait, really?
Yeah. Cancers secrete their own engineered exosomes into the microenvironment to deliberately force your macrophages into the M2 state.
Wait. The tumor wants the builders. Why would a tumor want a healing phenotype anywhere near it?
Because an M2 macrophage is heavily immunosuppressive. The tumor uses those M2 macrophages as like a localized shield to hide from T-cell surveillance.
Oh, why? So it's camouflage.
Exactly. And because M2s promote tissue building, the tumor exploits that to build new vascular networks to feed itself. It tricks the body into building blood vessels for the cancer.
That is incredibly sinister. So the tumor is weaponizing the healing process itself.
It really is.
That means flipping the switch is entirely dependent on the pathology. Like, we might want to force the M2 switch in ARDS to save a patient's lungs, but actively block the M2 switch in a tumor microenvironment.
And that is exactly why we have to understand the exact delivery mechanisms. If we want to engineer therapies, we have to know how these exosomes breach the macrophage in the first place.
Okay, let's unpack this, because how does a tiny lipid bubble actually get its payload inside a massive macrophage? Looking at the literature, it's not just a single entry point, is it?
No, not at all. There are three primary modes of exosomal signaling. The most direct is endocytosis, where the macrophage actively just engulfs the exosome into an internal endosome.
Basically swallowing it whole.
Right. And then you have membrane fusion. Since both the exosome and the cell have lipid bilayers, the exosome membrane simply merges with the macrophage membrane, and it just dumps its cytoplasmic cargo directly inside.
Okay, so no swallowing required there, just fusing and emptying the payload like a docking bay.
Precisely. But the third method is the one that really stands out. It's uptake-independent receptor binding.
Yeah, that one blew my mind. The exosome doesn't even need to enter the cell to reprogram it.
Right. It acts entirely from the outside. The exosome's surface proteins ligate-specific receptors on the macrophage membrane, which triggers an intracellular phosphorylation cascade.
So it just taps the outside of the glass and the whole inside changes.
Yeah, a classic example is exosomal CD47 binding to SIRP alpha on the macrophage. It transmits this definitive "don't eat me" signal, altering the macrophage's functional state without ever actually crossing the lipid bilayer.
This is where the bioengineering in these sources just becomes wild to me. Scientists aren't just observing this delivery, they are actively hijacking it.
Oh, the engineering is next level.
The papers describe metabolically engineering these exosomes using, let me get this right, axomanase and copper-free click chemistry. Which, I mean, it sounds completely sci-fi. How does that actually work on a molecular level?
It's a brilliant piece of bioorthogonal chemistry. Basically, you feed the parent cells, the ones producing the exosomes, an unnatural sugar called Ac4-mannase.
Okay, so they eat this weird sugar.
Right, and the cells process this sugar and incorporate these things called esoside groups into the glycoproteins on their own cell membrane. So when those cells secrete exosomes, those exosomes are automatically studded with these acis groups.
So they're essentially planting chemical anchors on the outside of the exosome, like little docking ports.
Exactly. Then the scientists introduce a targeted molecule, in this case, dextrin sulfate, that has been modified with a complementary alkane group.
Ah, I see where this is going.
Yeah, through copper-free click chemistry, the alkanine and the azide snap together seamlessly, like a molecular seatbelt. And it happens without needing any toxic catalysts, which is crucial.
Wow, so now you have an exosome perfectly coated in dextrin sulfate.
Yes.
And they do that because dextrin sulfate is a highly specific ligand for scavenger receptor class A, or SRA, right?
You got it.
And SRA is massively overexpressed on angry, pro-inflammatory M1 macrophages. So to use an analogy, it's like putting a highly specific shipping label on a package so it only gets delivered to the specific house on the street that is currently on fire.
That is a perfect analogy. The selectivity is the entire ball game in therapeutics. Because if you just blast the entire system with immunosuppressants, the side effects are devastating.
Right, you shut down the whole immune system.
Exactly. We see this level of precision with another strategy in the literature too, targeting the folate receptor beta.
Okay, why target folate receptor beta specifically?
Well, activated M1 macrophages upregulate folate receptor beta dramatically compared to just normal resting macrophages. So researchers took exosomes, loaded them with dexamethasone, a potent steroid, and functionalized their surface with folic acid.
Oh, that's clever.
Right. The engineered exosomes completely bypass normal tissue and home directly to the inflamed joints. You deliver a potent steroid exclusively to the cells driving the pathology, nowhere else.
That is incredible. So the package docks either by fusing, being swallowed, or binding to the surface like we talked about. Let's look at the actual software update happening inside the cell now.
The fun part.
Yeah. How does the cargo rewrite the macrophage's operating system? Because the sources constantly point to STAT3 as the master switch here.
Yeah, STAT3 is the critical juncture in the JAK-STAT signaling pathway for macrophage polarization. When the exosome delivers its cargo, it frequently alters the phosphorylation state of STAT3.
And phosphorylation is just adding a phosphate group to turn it on, right?
Right. Once phosphorated, STAT3 undergoes a conformational change. It dimerizes and translocates into the nucleus. And once it's in the nucleus, it binds to specific promoter regions on the DNA to transcribe M2-associated healing genes.
But the actual code on that exosomal USB drive driving this shift, it's mostly microRNAs, right? I was trying to visualize this. Like, how does a tiny snippet of non-coding RNA force a cell to completely abandon its M1 state? Because it's not building a new protein itself.
No, it's not building anything. It's actively destroying the M1 infrastructure.
Wait, destroying it?
Yeah. miRNAs operate through the RNA-induced silencing complex, or RISC. When the exosomal mRNA enters the cytoplasm, it guides this RISC complex to bind to complementary sequences on the macrophage's own messenger RNA.
Oh, I see.
Once bound, it basically cleaves the mRNA, preventing it from ever being translated into a pro-inflammatory protein in the first place.
Okay, so it's like a targeted assassination of the M1 blueprints before they can be built.
That's a great way to put it.
And the literature gives some incredibly specific examples of this too, like exosomes derived from bone marrow stem cells carry a microRNA called miR23A3P, and once inside, it specifically hunts down the mRNA for IRF1.
Which is vital, because IRF1 is a core transcription factor driving the whole M1 phenotype. By degrading IRF1 transcripts, the exosome essentially just removes the floorboards from underneath the M1 inflammatory program. The whole thing collapses.
There's another one that caught my eye too. Let7B5P. This miRNA targets and inhibits the TLR4 and NFKB pathway.
Yes, a very famous pathway.
Right, because NFKB is basically the primary alarm system for inflammation. So the exosome isn't just upregulating healing genes via STAT3, it is simultaneously snipping the wires of the cell's inflammatory alarm system.
You have to dismantle the old machinery to install the new. But this raises an important question, right? Biology is notoriously redundant. A cell will fight tooth and nail to maintain homeostasis.
Right, they don't like change.
Exactly. If you just inhibit one pathway, the cell usually compensates and finds a workaround. So how do these exosomes achieve such a robust, irreversible switch?
Well, reading through the data, they don't just hit one pathway. A single exosome payload is creating massive crosstalk across the cell. Like, they are hitting PI3KKT, the Notch cascade, and the WAMP pathway all simultaneously.
And that is the absolute key to exosomal efficacy.
Yeah.
A single exosome contains dozens of different miRNAs and proteins.
It's a cluster bomb.
Yeah, basically. By triggering PI3KKT to promote survival, while using Notch to shift differentiation and WANT to alter gene expression all at the exact same time, you create overwhelming regulatory pressure.
So the M1 macrophage just can't compensate.
No, it's totally boxed in. It forces a complete phenotypic overwrite to M2. There's no escape route.
Here's where it gets really interesting, though, because you can update the genetic software all you want, but to fundamentally alter how a cell actually behaves, you have to change how it generates power.
Metabolism.
Yes. The leap from genetic signaling to immunometabolism in these papers is just wild. The hardware power switch has to be flipped.
Yeah, we are increasingly realizing in the field that you just cannot divorce immune function from cellular metabolism. The metabolic disparity between M1 and M2 is profound.
Right, because M1 macrophages rely almost entirely on glycolysis, which, it makes sense in a way. It's fast and it doesn't require oxygen, which is perfect for an inflamed hypoxic tissue environment.
Right, but it is incredibly inefficient for the long term.
Yeah, but in M1s, glycolysis isn't just about making ATP, is it? The literature highlights that M1 macrophages have a fractured TCA cycle. And because that cycle is broken, specific metabolites accumulate, most notably succinate.
And succinate is fascinating because it isn't just a waste product. It becomes a primary signaling molecule itself.
Really?
Yeah, it literally leaks out of the mitochondria and inhibits prolyl hydroxylases in the cytoplasm. And that leaves HIF-1 alpha completely stabilized, which then translocates to the nucleus to drive massive production of IL-1 beta and other inflammatory cytokines.
So the altered metabolism is physically driving the inflammation. It's not just a side effect.
Exactly. Now, contrast that architecture with an M2 macrophage. M2s completely abandon glycolysis in favor of oxidative phosphorylation and fatty acid oxidation.
Right. They need that clean, sustained energy from intact mitochondria if they are going to spend weeks synthesizing collagen and remodeling tissue.
They need a bigger battery.
Yeah, like a hybrid car. The exosome literally forces the macrophage to switch its engine from burning gas glycolysis to running on an electric battery, which is OXPHOS.
I love that analogy.
And the research on adipose stem cell exosomes demonstrates this perfectly. These exosomes deliver a protein called IGF-2 directly into the macrophage.
Yep.
And that IGF-2 signaling cascade ultimately alters the cell's epigenetic landscape. Specifically, it changes the distribution of H3K27AC on the chromatin.
Which is huge.
Wait, so it's actively acetylating the histones?
Yes, it physically opens up entirely different regions of the DNA for transcription, specifically committing the cell's metabolic grid to oxidative phosphorylation. It rewires the power infrastructure at the fundamental epigenetic level.
That is crazy. And then you have the bone marrow stem cell exosomes hitting it from the other side. They actively throttle glycolysis. The data shows they reduce the phosphorylation of P65 and P38, which just plummets lactate production, and they simultaneously restore the mitochondrial membrane potential.
They basically go in and fix the fractured TCA cycle so that succinate stops accumulating and driving the fire.
But again, like we mentioned earlier, this machinery is easily weaponized. Tumor-derived exosomes exploit this exact metabolic vulnerability.
Oh, absolutely. They don't just send genetic signals. They dump raw metabolic enzymes into the macrophages.
It's so aggressive.
It really is.
Yeah.
Tumors will pack their exosomes with hexokinase 2 and just massive amounts of lactate, intentionally flooding the macrophage's cytoplasm to forcefully skew its metabolic state.
So they are literally force-feeding the macrophage a specific metabolic diet to brainwash it into becoming an M2 bodyguard.
Yes.
That is genuinely unsettling.
It is. But if we connect this to the bigger picture, the paradigm shift in these papers is absolute. We traditionally viewed immunometabolism as a trailing indicator. Like a cell gets inflamed and its metabolism adjusts to meet the new energy demands.
Like it was just reacting.
Right. But the causal arrow actually points both ways.
The metabolic state dictates the immune phenotype.
Exactly. The sources point out how some exosomes deliver SRT1, which is an enzyme that acts as a direct cellular energy sensor.
Okay, so what does SRT1 do once it's inside?
SRT1 enters the macrophage and deacetylates a protein called FXR, and that deacetylation event physically shuts down the NLRP3 inflammasome.
Wow. So you are directly linking a metabolic energy sensor to the physical protein complex that initiates inflammation.
Yep. If you control metabolism, you entirely control the macrophage.
The implications for therapeutics here are just staggering. I mean, we are moving way past the era of injecting whole stem cells and just hoping they engraft somewhere useful.
Right, because whole cells are unpredictable. They can differentiate into things you don't want.
Exactly. But with exosomes, we have a cell-free therapy. We can harvest these vesicles, use click chemistry to bolt on targeting ligands like dextrin sulfate, load them with miR-23A3P to silence the M1 genes, and pack them with IGF-2 to force oxidative phosphorylation.
We can engineer the perfect biological payload.
And the targets for that payload are some of the most intractable diseases we face. If you look at severe infections or acute lung injuries where ARDS develops, the mortality is driven by M1 macrophages, basically stuck in a destructive loop, essentially dissolving the lung architecture.
Or in osteoarthritis, where those same M1 macrophages are endlessly degrading articular cartilage in the joints.
Right. Being able to definitively flip that switch to M2 to halt the destruction and command the cells to rebuild the matrix, it just completely changes how we view chronic disease.
It really does.
Yeah.
It gives us a programmable intervention at the literal molecular level.
So what does this all mean? We are looking at a near future where we can reprogram our internal cellular machinery basically on demand. But taking a step back from specific pathologies, there is an overarching concept in the literature regarding how we age overall.
Ah, yes.
Because a primary driver of biological aging is chronic, systemic, low-grade inflammation.
Right, what we call inflammaging.
Yeah.
As time passes, we accumulate this diffuse, just stubborn population of M1 macrophages across all our tissues.
Just everywhere.
Yeah. They get stuck in the on position, generating a constant hum of reactive oxygen species and cytokines that slowly degrade tissue integrity over decades.
Which sets up an incredible proposition for the listener to think about. If we truly master this exosomal M1 to M2 switch, if we can engineer these lipid USB drives to seek out and reprogram those stubborn inflammatory cells with perfect precision, could we eventually deploy them to not just treat a localized injury, but to systematically flip that switch across the entire body?
That's the million-dollar question.
Right. Could we use these tiny vesicles to halt, or potentially even reverse, the fundamental cellular wear and tear of aging itself?
It is the absolute frontier of longevity research. I mean, if you have the molecular tools to command the builders, to clean up the damage, restore the mitochondria, and just stop the fire, you are essentially rewriting the biological clock.
It's incredible.
The exosomes possess the exact instruction manual for cellular rejuvenation. We are simply learning how to type in their language.
It fundamentally changes how you view your own immune system. It isn't just a static defense force, it is a highly programmable network, just waiting for the right code to be uploaded. Well, thanks for joining us on this deep dive. Keep asking the big questions, keep questioning the mechanics of how it all works, and we'll catch you next time.